Yep, okay. Well, welcome to our second PACVEC seminar. I guess this is the first research one, and thank you to Sarah here and the team at Davis for helping us to get the system coordinated in the last 60 minutes. So this is good news. So I'm going to talk to you today about a system that we've worked on for several years. It's called the piRNA system in the yellow fever mosquito Aedes aegypti, and its possible role in transposon silencing, which is no surprise if you are familiar with this small RNA system, but also, as we've recently proposed, at least in the lab, perhaps a segue into a new system of immune priming. And so the outline of the seminar will be that very quickly I'm going to talk about the genetic tools that are now available in insects, but we're primarily talking here about mosquitoes and Aedes aegypti. I want to cover why we're investigating the piRNA pathway to begin with. I want to talk about the reason we got into the piRNA pathway with respect to Aedes. And then we'll talk about some early studies that a previous graduate student did called Jennifer Wright, who actually was a Davis graduate, I think coming from Greg's lab. And then most of the work, or most of the talk, will be on a recent student from the lab who defended successfully last June called Michael Ham, who really started then to do some, I think, nice work on shedding light on this system. I'll then finish with a model. I'll talk about where these data fit within this small emerging field, and then conclude with ongoing research. And I'll keep my eye on the time so that I might skip some things if I see myself running out of time. So since 1997 or thereabouts, when repeatable genetic transformation was achieved in mosquitoes, Aedes aegypti to begin with, we've seen a gradual increase in the number of generic genetic tools that can be harnessed to study the genetics and biochemistry of mosquitoes. So it started with class 2 transposons, initially Moz and Hermes in Tony James's lab at Irvine, and then Moz and Hermes, that's correct, and then followed in short suit by Piggyback and Alex Reichel's lab at Michigan State, and then later on TC Buster and also and Muta-1, which is some unpublished work in our laboratory. And transposons are good. They are still used to actually transform mosquitoes, even in the CRISPR era. And certainly for many applications of CRISPR technology, you still need a way of integrating some components of it into the genome if you want to do more sophisticated genetics. One of the weaknesses of transposons is that you don't have any control over where they integrate. And even though I've listed perhaps 8 of them there, they have a spotty ability to function. So I've put up Minos there. Minos functions well in anophelines, Anopheles stevensii, but does not work at all in Aedes aegypti. Site-specific recombination is another generic tool that's available to us. It's a system that's from bacteria and fungi. There's some of those common ones used now, particularly the STI-C31 system. RNAi has been used spottingly in mosquitoes and other insects. In some cases it works quite well, in other cases it doesn't. And even within mosquitoes, within individual species, it can vary tremendously depending on when you do the injections and where you inject. And then quite recently, since 2013, 2014, there's been a revolution in CRISPR technology. You know the story, I don't tell you the story, but pretty much every organism that folks try and get this to work in, it works. And one of the beauties of this is that in its most simplest applications, you don't need transposons, you don't need endogenous promoters. If you just simply want to make mutations, you can just inject the components of CRISPR-Cas9 into your organism, and it works for you. And one of the biggest challenges now that remains for truly exploiting this technology is DNA delivery. So in mosquitoes, because we know we can inject into pre-blastoderm embryos, that's fairly straightforward, but in other insect species, it can be. And then last, promoters and gain of function dominant genetic markers. The dominant genetic markers have been around since the discovery and use of DFP and other fluorescent protein markers from the early to mid 1990s. But as these genetic technologies that are listed up there continue to grow, one of the problems we run into, not just in mosquitoes, but in other insects as well, is tissue-specific and temporal-specific promoters. And so even if you look at simple promoters like a synthetic promoter like 3xp3, which works well in Drosophila, and some other species like mosquitoes, for example, for reasons unknown, it doesn't work in typical fruit flies. And so there's a greater emphasis coming on identifying specific promoters from specific species to work in a particular tissue. And then all of these components can be folded into a system, into various systems, and I've just mentioned some of them there, gene and enhancer trapping systems, recombination medium cassette exchange, which themselves can be folded into larger systems, for example, with RMCE using CRISPR-Cas9 technology. So this is a great time to be an insect geneticist. You can get away from Drosophila. You can now start deploying all these tools in your insect of choice. Most of the things that I just showed you on that previous slide were generic, with the exceptions of some of those transposons, with the exceptions of promoters. So why did we get interested in piRNAs? Because when we and others started to work with Aedes aegypti in the late 1990s and early aughts to put genes in, the idea was, way back then, that perhaps transposons could be used as gene drive. That idea evaporated pretty quickly in the mid to late aughts as Wolbachia came online. And as a concept, it's pretty extinct now with gene drive and CRISPR-Cas9 going on. But we were interested in that, and we're also interested in developing genetic tools like a gene and enhancer trapping in Aedes aegypti. And so our group and others listed here found out pretty quickly in the mid to late aughts and early teens of this century that Aedes aegypti, if you will, was an insect where transposons went to die. And so even though David O'Brien with some exquisite work after he failed to get any gene or enhancer trapping working in Aedes aegypti in the late aughts and early teens, then moved the same plasmids, the same cassettes he was developing for Aedes aegypti, which failed, into Anopheles stevensii, and was very quickly able to get gene and enhancer trapping working in Anopheles stevensii with a piggyback-based system. But in fact, he was able to show in these quite elegant experiments, the second reference from the bottom, that piggyback, which can be actually remobile, which can be active somatically, doesn't even work somatically in Aedes aegypti. It's as if there was something really suppressing transposons in Aedes aegypti. So at that same time as these experiments were starting to go south, not just in our lab, but in other labs as well, Julius Brennicke and Greg Hannon's lab at Cold Spring Harbor discovered these new piRNAs. And they were simply a new type of small RNAs. We were used to siRNAs that are involved in antiviral resistance in vertebrates and invertebrates. We were also used to microRNAs that are small RNAs involved in gene regulation. They discovered this third pathway, and very quickly they did some very elegant experiments using the whole genomics approach and some genetics, which showed that piRNAs in Drosophila melanogaster, and most likely in mice as well, were responsible for suppressing transposon movement across a genome. So those of us who had studied transposons for a long time were certainly aware of exquisite local transposon regulation systems. So the P element at that time was one example of that. There were other mechanisms, especially in prokaryotic transposons, showing how a particular transposon could auto-regulate itself. But there was always a feel that there was a generic genome-wide system that could recognize any of these types of transposons and somehow inactivate them. And basically what Hannon, Brennicke, and his group did in the late aughts and early teens, and it continues on to this day, was to understand how the piRNA system would silence transposons in Drosophila. And other groups were doing the same in mice, including theirs, and also to a lesser extent in zebras. So we became interested in trying to see if that was the case in Aedes aegypti. Now, straight away, we ran into a problem, not a problem, but just a change in that, as had been shown by Corey Campbell at Colorado State at about the same time we were running the QLEX genome project, she found that there was an expansion of piRNA biogenesis genes in Aedes aegypti. So if we looked at the Drosophila work, we would have three major piRNA biogenesis genes: AGO3, piWE, and albumin. And you can see from this chart that they have those Drosophila ones circled, and you can see how this is a phylogenetic tree. And then you can see that in mosquitoes, and just look at Aedes aegypti, which is in the magenta, you can see whilst we have a single AGO3 gene, which is mapping closely to the Drosophila AGO3 gene, we have 7 piWE genes. And so there's, on the face of it, an initial complication. And I want you to remember that Drosophila, piWE, and albumin are close to each other. As you'll see in a few moments, Drosophila piWE is involved in what we call primary piRNA pathways. AGO3 and albumin are involved in the secondary piRNA pathway. And very quickly, when we did some bioinformatic analysis, we concluded that the single Aedes aegypti AGO3 gene was most likely corresponding to the Drosophila AGO3 gene, and most likely the piWE2 gene of Aedes aegypti corresponded to Drosophila albumin, at least bioinformatically. So this is I hope you can see the references, and I'm referencing these figures that I've taken from other papers. It's not coming up on this screen, but hopefully it's up on Davis's. So this is just two diagrams from a very recent review by Sayoki, who's one of the pioneers of the piRNA field in Drosophila. And I'll just tell you now that it's got a lot more complicated than in the 2007 Cell paper from his lab. So whilst I talked about AGO3, piWE2, and albumin, there are many, many more partners now involved in this. And I'm not going to spend this seminar going through these slides. Suffice to say, there are two pathways in Drosophila. There's a somatic pathway on your left, and there's a germline pathway on your right. Somatic pathway still involves only piWE. And in Drosophila melanogaster, the somatic pathway is totally confined to one somatic tissue, and that is the follicular cells surrounding the ovary. There is no somatic activity that anybody has characterized elsewhere in the insect. And that is driven by the piWE gene, which basically is processing the piRNAs that are coming from these large unidirectional clusters. And then those piRNAs, after processing, to keep it very simple, go back into the nucleus and silence transposons. If that transposon is pre-existing in the nucleus, then if a transposon that's identical to it invades, it is shut down by virtue of this pathway. There's a second pathway in Drosophila and in other insects, including Aegypti, called the secondary piRNA pathway. And that's specific to the germline, so that's specific to the true germline, not the follicular cells around the ovary, of both in the ovaries and in the testes. And that also includes components of the primary pathway, but it also includes a secondary pathway, which has a so-called amplification cycle, which basically increases the level of small piRNAs that are produced to the original piRNAs that are transcribed from the nucleus, and if you will, amplify or up the response. Now, this occurs in a structure called the nuage, which surrounds only germline nuclei, and the idea is that it's protecting the germline nucleus at the most sensitive time prior and during early development, when you can't have things, if you will, going awry through transposons jumping around. So that is very complicated, and that's a paper that, as you can see, the references from Sayomi. But for the purpose of this talk, we're just going to keep it simple, because our exploration into the piRNA system in Aedes is still very rudimentary. So somatic, it's primary piRNA. The piRNAs are 23 to 30 nucleotides long with a U1 bias. In other words, we tend to see uracil predominantly at the first nucleotide. There's one key piRNA bias, it's gene in Drosophila, that's piWi. There's no amplifications, and the piRNA clusters are unidirectional. That means only one strand is read. And I won't read out what is on your right, which is just simply the differences with the secondary system. So that's the whole thing simplified, and that's all we need to know moving forward. So early on, Jennifer Wright and myself asked this question in piRNA 2011, what do these genes look like in Aedes aegypti? And we were just simply trying to find gross differences in this very early gel. And the take-home message was this, that the majority of these genes, virtually all of them, were transcribed, and you can see them there in the eggs, in ovaries, and a carcass. So that's carcass that does not contain any germline tissue at all. We knew that piWi1 was simply a deleted version of a deleted form of piWi2, so we didn't include it here. And what we concluded from this and some other studies is that we had to be very selective at this time in the lab, was that we're interested in piWi2 because it was hardly expressed in carcass. It was only found in the eggs and in the germline tissue. And we'd already shown that it was probably identical or orthologous to albumin. And we're interested in AGO3 because it was also had a fairly significant role in piRNA biogenesis. But it was clear that these other piWis, other than piWi2, were also expressed in somatic tissues, which was very different than the situation in Drosophila, where the only somatic tissue is follicular cells surrounding the ovary. Here, at least at a very gross level, we found piRNAs being made elsewhere. And in fact, in Drosophila, it's only piWi itself that's in somatic tissue. Everything else occurs in germline tissue. So straight away, Aedes aegypti was different. These 2011 results were subsequently confirmed by other groups working on mosquito cell culture in Scotland and in the Netherlands. So we confirmed this strangeness of AGO3 and piWi2 with RT-qPCR, and you can actually see with piWi2 there's expression in the ovaries and early eggs before the zygotic genome kicks in, and for AGO3 we can see strong expression in the carcass. So that was reassuring. And subsequent to that, Omar Akhbari, not interested in this pathway, but doing a very comprehensive RNA-seq analysis of every gene in Aedes aegypti. This was published in Gene 3 in 2013. Every gene was examined. We're just interested in these. And this also confirmed, albeit low levels, AGO3 expression in carcass. piWi4, 5, and 6 expression also in carcass tissue, confirming Jennifer Wright's original results. And then piWi2 was actually down even in the germline, at least in 12-hour blood fed ovaries, there was some interest, or some level of expression. So we undertook then two approaches to understand how piWi2 and AGO3 would be working. First is that we wanted to make polyclonal antibodies specific to each protein using a unique peptide sequence at the carboxy end of each of these genes, proteins rather. And then we wanted to actually make some transgenics and use snapback sequences to see if we could get knockdown sequences and see if we could get a loss of function. This was pre-CRISPR days. CRISPR was certainly being discussed and examined in bacteria, but it hadn't become a genetic tool that it is now. So we started with co-immunoprecipitation. So we just took blood-fed ovaries. We found either the piWi2 antibody or the AGO3 protein, and then we basically purified the library, the piRNAs that we pulled down with those. We eliminated siRNAs and microRNAs, which are 21 nucleotides in length. We did that bioinformatically, and we did sequence analysis. One thing which petrified us at the time, and also continues to worry me, because I have to keep worrying, was, how specific were these antibodies? The AGO and piWi proteins are related to each other, and even though we took a specific peptide sequence at the carboxy end, there's always this risk of cross-contamination or cross-reactivity with these antibodies. So we did some mass spec analysis. We did Western blot analysis with the piWi2 antibody when these came from blood-fed ovaries, so you'll see the collagen, actin, etc. We certainly recovered piWi2 and, unfortunately, piWi3 from the PIWI2 pull downs, and as you'll see as this talk goes on, only AGO3 from the mass spec analysis of the pull downs of the AGO3 protein, the collagen and actin, etc. So fairly comfortable with that, especially with AGO3. So when we looked at the libraries that were pulled down by these proteins, not surprisingly, we find a strong transposon complement shown in the pale blue sectoring there. But as we had found earlier when we published this work with Peter Ahrensberger, who was a postdoc in the lab in 2011, one of the surprising things about Aedes aegypti, unlike Drosophila, is that many of the piRNA targets go to genes, and some of those are viruses. So this was not surprising, but it made us become a little more interested in these genes. And so we started to do immunocytochemistry, and this is where Michael Hand's work now kicked into the rest of the talk. And so this is just a diagram from Julius Brennicke's landmark paper in 2007, showing the localization of piwi, aubergine, and AGO3, which the audience here can see, this is AGO3 in the developing ovaries of Drosophila melanogaster. And we're just going to focus now on piwi2 and AGO3, but I'll tell you now that piwi2 is found in the nuage, which is that structure which is around the germline nuclei, which is thought to act as a gatekeeper around the nuclear pores. And at least in Drosophila melanogaster, we know that in the nuage it partners with aubergine to basically do this ping-pong amplification cycle. And as I said, AGO3 was also involved in the nuage. But Michael found something very different. So if we looked at piwi2, which is both here and in the middle and low down, most certainly you can find that association in the nuage. The next slide, which is a close-up, will actually further emphasize that. But what we found with AGO3 was quite surprising, because here you can see, especially on that image on the top panel to your right, you can see the DAPI staining is staining the nuclei quite well, but AGO3 is actually in the follicle cells. It's in the cytoplasm of the follicle cells. You see that clearly in the next slide. And it's also dispersed in the through the cytoplasm in the nerve cells. That's quite different than what we see for AGO3 in Drosophila. So here's just simply a close-up of AGO3 in piwi2. You can see on the right-hand side, piwi2 is very nicely localized in the nuage that's surrounding the germline nuclei. And that's pretty much identical to what is seen in Drosophila melanogaster. But AGO3 is certainly absent from the nuage. So if you look at those 3 or 4 nerve cell nuclei, you don't see any signs of localization or redness around the nuclei. You can see smidgens in the cytoplasm, but you can certainly see it extremely well in the cytoplasm of the nuclei, in cytoplasm of the follicular cells. It's also present in the testes of Aedes aegypti, as is piwi2. If you want to take a real close look, you'll see that it's not present in nuclei. It's not as clear as what you can see in nerve cells. And so, just to simply conclude this section, we found something different about AGO3. We're convinced, as we can be from Western blots, from the analysis of the mass specs, that this is AGO3 and from its sequence analysis. But it's behaving a little differently than what we see AGO3 in Drosophila. And maybe that's not unusual. Maybe that's consistent with what I've shown you previously, which is a larger somatic footprint of AGO3, not just in germline tissues and somatic tissues associated with the germline, but in the rest of the mosquito. Now, if we wanted to look at the Drosophila model, that presents us with a problem, because straight away we have to ask the question, well, if it's not in the nuage, what is it partnering with anything else? In Drosophila, AGO3 partners with aubergine, and you can read what I've put up there on the slide. Suffice to say, understanding what those partners would be in Aedes aegypti is an interesting task. And because we know we have an expansion of the piwi genes, we have 6 functional piwi genes as best we can tell, any of those others, piwis 3, 4, 5, 6, maybe 7, would be good candidates. And in fact, we've already got evidence now later on, which I'll show you, that it could be 4, 5, or 6, which is supported by independent work that is going on in the Netherlands in cell culture, but not in whole mosquitoes. So in the piRNA mosquito world, which is extremely big, tiny, we're the only group that's actually looking, as best we know, in the whole insects and not in cell culture. So let's just skip that. So that was our first pass at immunocytochemistry. I'll come back to that in just a moment because I have some more images to show you, but it's a very different issue. The other approach we took to interrogate this system was generating transgenic mosquitoes that had snapback sequences. So this was a plasmid that Rob Heiss and I designed, and Rob made when he was an SRA in the lab before he retired 2 or 3 years ago. And basically it uses the TED on-off system. So in mosquitoes, you can rear larvae in water and add tetracycline at a known dose at a known larval density. And the idea here was that we would put in a snapback sequence to PW2 or AGO3. And the idea would be that when you grow up the larvae in tetracycline, you would get reduction of that particular snapback, and that would silence in the perfect world, would silence that particular piRNA biosynthetic gene, and maybe that would lead to a de-repression of transposons, if in fact piRNA is working. So we made those mosquitoes. Despite all our efforts, we were never able to get a PW2 snapback mutant. We've interpreted that is either complete incompetence on our part, or most likely because albugin's many alleles in Drosophila albuginea lethal, maybe PW2 when you inactivate it as lethal. But we could never get anything. So I don't think we're incompetent because we could get the AGO3 version. We can generate transcripts doing that since 1999. So we obtained several lines containing the AGO3 snapback PW transposon. So at this point, the analysis of PW2 stops, except for some immunocytochemistry I'll show you in a moment. So we generated several lines. The female lines that were generated, when I say female, they were initially found in a G0 female, did not prove to be of any real interest. But we found one line that straight away, when we saw that as G1 individuals, G1 transgenics, had a strange longevity issue. So it just seemed to be slower or faster. And so once we had that homozygosed, homozygosed, we started to look at this. And so here we have, we're going to call this line M14. And here it is in this table. So you can see the hatch rate. I could say 49%, but I wanted to show you here that when we say 49%, we looked at 100 larvae as opposed to looking at 10. Right? So that's why it's not as a percent. And you can read that table, and you can see that when we induce A3 snapback, so that's larvae grown in tetracycline versus larvae from the same strain not grown, and then there's Orlando is just a wild type strain that that are the background strain for the mutations. And you can see that there's a lethality and longevity issue. You can see that in M14 males grown in the presence of tetracycline, they get stalled at late 3rd and 4th instar. And in fact, they die, whereas the females are fine. And I don't have, and will not have an explanation for that, beyond a role for AGO3. Okay? So this line is now many, many years old and maintains this thing. So that was curious, and it actually started, and we looked at various things to say, is this trivial? Is there other things going on? But we couldn't find anything that offered any other explanation other than it was something that potentially AGO3 was doing. And we'd never looked at 4th instar larvae before. I mean, certainly in Drosophila world, it's a 3rd instar is the equivalent. In other words, it's a final stage before puparation. I mean, why look? It's of no genetic consequence at all. So we were interested in that. And we confirmed that in the M40 induced males, we had a de-repression of AGO3, which is pretty significant. We also found that if we relax selection, in other words, when I say selection, I don't mean tetracycline because that's an inducing treatment. I mean, originally these were found by looking for GFP in the eyes, which was a 3xP3 GFP marker. when we relaxed selection after many generations, we found that we've got a new phenotype in the males, not shown well here, but it was basically green. This is a pupae, one pupae here, another one here, and they're inverted to each other. Here's one eye, here's the other eye. We basically saw green fluorescent legs and some markings in the abdomen. And females lost GFP altogether in the absence of looking for GFP in the eyes. But the females actually still retained the gene. We don't have a good explanation for this other than the observation is that in the relaxation of selection, that particular genetic marker, which is the eye color, we seem to have some suppression going on in the females, but not the males. But when we maintain selection for eye color, all the phenotypes that I previously described are delayed development of the problem. So we looked at where these integrations were. One possible explanation for this strange phenotype is that it has nothing to do with AGO3. It just hit a gene which is important. And so we had to find out whether that was true or not. We found 3 integrations of this piggyback snapback AGO3 transposon in the genome. You can see them there. The first one was in the 3' UTR of a gene orthologous to the meiotic P26 gene, which, as you can read, basically does present fertility problems in both sexes, but not lethality, Drosophila melanogaster. This is not in the coding sequence, it's in the 3' UTR, and RNA-seq analysis indicates there's no difference in transcriptional levels of this particular gene in this strain of aegypti. So as best we can tell at that level of analysis, there's no effect on MyP26, the orthologous. And then you can see for the other 2 that they are at least 70 to 100 kilobase away from any known gene. We look for microRNA genes, any other non-coding sequences that could be of interest, but as best we can tell, these last 2 sites are effectively neutral in terms of gene function. So we started to look at, is it artifact? Is it real? So we started to look at 4th instar larvae. Is it telling us that there's something special about 4th instar larvae in Aegypti? And as I said, if you look at this in Drosophila, you have no reason to ask those questions because of the confinement of the piRNA pathway to the germline and the ovarian follicular cells. And so here's an image of a larvae that I've just pirated from a paper by Linster and Ding Larsen recently. And those of you who do mosquitoes will be familiar with this, with the figure down the bottom. And what's peculiar about 4th instar larvae in Aedes aegypti in the context of transposons, which is where we're coming from, what is silencing transposons in Aedes aegypti? Well, there is a rapid rate of cell division in the gonads. And so one might expect a priori that there could be increased transposition if that cell division has been accompanied by increased transcription in the gonads. And the other thing about pupae 4th instar is that that's the maximum level of feeding before it forms a pupa. So that's when the feeding is its most voracious. That doesn't seem to have anything to do with transposons. So Michael Hahn, with all his data, asked the question whether there really was AGO3 enrichment. In other words, if you looked at the expression of AGO3 in 4th instar larvae and then dissected our tissues, in this case, we were just very blunt with our tissues. We just took the head, we took the gastric sichi, which is inevitably interwound with the salivary glands. So I'm just going to, when I say gastric sichi, I am saying salivary glands by default because it's very difficult to separate them. And then the rest of the gut. And what he found was that, once again, comparing to the levels of AGO3 across the 4th instar larvae, this is done several times, you can find that there's a significant enrichment in the gastric sichi / salivary glands, not in the head, not in the rest of the gut. And then the outlier up here, the log scale here on the X is the expression ratios, the reference like it's just blood-fed ovaries. So it's still way down what you see in blood-fed ovaries. Let's not start confusing our priorities here about what piRNA is doing. I mean, just as you see in Drosophila, presumably in these developing ovaries, its prime function is to silence transposon. But what we are seeing in Aedes aegypti is expression in this unusual tissue. unusual in terms of thinking about piRNA. So I promised to come back to immunocytochemistry. And so here is now dissected salivary glands gastric siki from Aedes aegypti, showing expression of, enhanced expression, localization, I should say, of AGO3 in the middle portion of the top panel to salivary glands / gastric siki. What you're really seeing there is gastric siki. The salivary glands are quite tiny in comparison. And then the second one is simply an EGFP control, and the last is AGO3 from the larval height. So DAPI staining in the first row, AGO3 in the second. Right. So once again, this is supporting the RT-qPCR data, showing that AGO3 is present there. So that is encouraging, but we still don't know what it's doing. So what we also wanted to look at, what was driving the study early on, and this is just simply that we're becoming less interested in transposons in some sense and more interested in other things. This was all about looking for de-repression of transposons. So early on, and now we're looking at fourth instar, we're sort of saying that, looking at transposon de-repression in ovaries is probably going to give you the results you expect, right? So even though we've done that, I'm not showing this here, results are all the same. But here we selected 6 transposons randomly, although semi-randomly. These Buster-1 mutator ones are ones that we've isolated in the lab. They're class 2 active transposons from Aedes aegypti, and we also went into class 1 transposons. And so AGO3 is showing decrease in RNA-seq, which is to be expected. These are transcriptional levels, not piRNAs. So our AGO3, as you expect, will be significantly downregulated in these snapbacks. And here you have, these are 2 transposons that are significantly upregulated. Now, when we look at the entire transposon complement, so we just were sort of semi-random with 6 transposons. But here are all the transposons that are significantly expressed. By significant, they have detectable levels of transcript. And this is M14 male versus an M14 female uninduced. We don't expect to see any change. And in fact, I can't ask you to look for the red dots, but I can assure you that where we have red dots, we have significant changes in expression. And over here in the males, which is the panel to the right, all these outliers. And also be aware that the y-axis has changed considerably between females and males. We have a whole series of transposons that are deregulated. What are they? Most of them are class 1 transposons. Most of them are gypsy elements. Gypsy elements are interesting transposons for 2 reasons in this study. One is that if we go back and look at those major piRNA loci that Brennicke found in Drosophila, especially in the follicular cells, it's gypsy transposons that most of the piRNAs are going to. The second observation about gypsy elements, which is going back to some work that Alain Boucheton did in France many, many years ago, is that gypsy elements can be infectious. If you grow up Drosophila that don't have gypsy, in media that contains remnant, mashed up DNA from gypsy elements, they can actually get infected. So the gypsy elements are sort of bordering this line between transposons and viruses. And of course, many, especially class 1 transposons, the retrotransposons are of course derived from viruses. We can also look at piRNA levels. And once again, these are from just males in this case. You might see that there are a whole pile of red dots under that line, which are piRNAs that are targeting transposons that are significantly switched down in the AGO3 mutant. So these last few slides confirm, and here are their targets. And once again, gypsy elements are predominant. Lastly, in this particular section, we're interested in what may be partnering AGO3. So if AGO3 is active somatically in this particular tissue, 4th instar, gastric siki, does it have a partner? Recall the earlier slides of AGO3 partnering with algae in the nuage of Drosophila. Well, does AGO3 have somebody to dance with in this particular tissue? And so Michael then looked for which PLE genes are significantly upregulated in gastric siki, salivary glands in the 4th instar, not PIWI3, but PIWI4, 5, and 6. And this is supported by data from Van Rij's group in Holland, looking at Aedes aegypti cell culture, which has also shown elevated levels of these particular genes and AGO3 in Aedes cell culture. So we started asking the question of what could be going on. You know, what's special about gastric siki? As much as I showed you transposon data, and I started my research in this field on trying to find new transposons that could transform insects, it's not that interesting here. I showed those data to you to support the notion that the experiments we started to do, which was to generate mutants of Aedes aegypti that would knock down a piRNA biosynthetic gene, would silence transposons through a loss of piRNAs. And I've sort of shown that that's going on. But that's not really intriguing. It's to be expected, albeit in a very different tissue at a very different time than Drosophila. We started to think, what's going on? What the gastric siki is not contributing to the germline. I think we can be fairly certain of that. If that's not the case, then speak up now. So what is it? Well, the gastric siki is housing many bacteria. I mean, as the larvae is feeding, everything that it's taking in initially is going through the foregut, the gastric siki. And what if it's encountering pathogens, whether they're viruses? Viruses, of course, are extremely exquisitely related to transposon. And what if somehow, and we'll get to the model later, that's got something to do with piRNA biosynthesis, the selection for it, the fourth instar larvae. So one thing to note here is that, so these are the top 30 reads from gastric siki and salivary gland libraries generated in the lab, mapping to viruses. And notice that, of course, most of the double-strand DNA. The piRNA system is a DNA system. Even though it can target retrotransposons, those retrotransposons in Drosophila and other organisms go through a DNA intermediate which sits in the nucleus, so it can live with it. You will see in one case we have double-strand RNA leads. It's now Ning virus. And we also have in the next slide, which is from the same but from whole larvae, the radioviruses as well. And the radioviruses, and also I should point out, which Lee and I were sort of reading about the other day, these mimiviruses from amoeba, these amoeba are also digested by mosquitoes. Now, let's be clear here. Some of these numbers are tiny. These are the counts. I mean, we're not getting too carried away here. We're just simply looking for an explanation. But it could also be, remember, these mosquitoes are reared in the lab. These have been reared in the lab before my time here. I mean, I think I got this strain from you, Brian, Orlando. Was here in '97. So it's not as if we just hauled these from a pond that Bill goes to every week. These are lab-bred mosquitoes. So these are in the genome. I want to get back to that in just a moment. So the summary of somatic expression is that we're able to obtain PIWI snapback mutants. We found basically a strange phenotype, which we still believe is due to AGO3. That keyed us into looking at fourth instar larvae. And we can see a repression of transposons through knockdown of AGO3. We can see a reduction in piRNAs targeting transposons, and as a result, increase in transcription from transposons. I'm not going to get time to go through the piRNA architecture because I want to get to the model. But suffice to say, as I just flick through these slides, and I'll just stop at that one. Remember from that simple slide, somatic is unidirectional clusters, germline is bidirectional. By and large, the piRNA clusters that we see in gastric siki, fourth instar, are unidirectional, consistent with what happens in Drosophila and follicular cells. What we did see, which is different, was that some small sign of piRNA. So to very simplify this very greatly, those top two slides, the top two A and B are 5' overlaps from Aedes and Drosophila. Aedes, ovaries, and Drosophila. And I was talking about that ping pong amplification cycle. All you need to know is that there's a 10 base pair overlap which drives that cycle. I don't have time to go. You can see that that's the case in Aedes aegypti, in ovaries, and also in Drosophila. But when we look at gastric cechi and we look for that 10 base pair overlap, we can see that there's some evidence for it. So it's fourth instar in C and libraries from gastric cechi in D. But you can see it's sort of weakening, or at least there's some perturbation of it. And that hasn't been seen before in any other organism in which piRNAs have been found. I should also say that the clusters we see in gastric cechi, the piRNA clusters, are unique to gastric cechi. We don't see those expressed in the ovaries or in the embryo. In fact, what we're seeing in Aedes aegypti is a progression of piRNA regulation through development. So it's not as if here are your piRNA loci, they're switched on all the time. Different piRNA loci are switched on at different times. But I want to get to the model for the brevity of time. I should also point out last thing before I get to the model. Previous to Michael's work, there were two known types of architectures of piRNA clusters. One showing unidirectional at the top, and it can be unidirectional in this direction or unidirectional, Davis, in the other directional, but not both. You can have bidirectional. And so these are typically found in the germline. These are typically found in soma. And then what Michael found was a new type of cluster, which is bidirectional, which has transcription from here and here, and absolutely terminates at this point. And in fact, many of the piRNA clusters he found look like this. And we're interested to know what the sequence, what is the actually, is there a sequence here that is such a potent transcriptional terminator. Let's get to the model. So this is our model. And this is, it should be challenged because it's a model. So we're proposing that for larvae, if the fourth instars are feeding voraciously, of course they're fed as first and second and thirds, but they're feeding very, very at high levels. They take up a virus, and maybe some of these viruses, like iridoviruses, are pathogenic to mosquitoes. And so in some cases, the mosquito dies. In other cases, and the thickness of these arrows show perhaps the frequency of events or the likelihood of events, you get a non-lethal infection. So the mosquito lives, and as we know, during metamorphosis, there's complete puparation, there's complete metamorphosis. And this is what usually happens: death or survival. No impact at all on piRNAs. But maybe in some cases during metamorphosis, it's a very rare event. And when you work on transposons invading organisms, you get used to rare events. P invaded Drosophila in the New World 80 years ago, and now, 80 years later, P elements are found. It could be one rare event. Maybe sometimes during metamorphosis, that particular DNA virus integrates into the genome. Right? Tissues are broken down, tissues are reformed. It doesn't have to be frequent. And maybe most of the time it doesn't go into a piRNA locus. So it's neutral, nothing happens. But maybe sometimes it goes into a piRNA locus. And maybe sometimes it goes into a piRNA locus. It's expressed in gastric cechi. And maybe sometimes when that happens, it's affording resistance against that particular DNA pathogenic virus, in which case there would be selection for that individual. So that's the model we're working with. The context of our data and model in the field is that since we started this work with Jennifer and Peter Arensberger and published, several groups have worked exclusively on Aedes aegypti cell culture and have come up with data which are supporting our observations of the somatic expression of AGO3, PIWI4, 5, and 6 in cell culture. They have found that when they infect cell culture with flaviviruses, that an siRNA response is activated, as is a piRNA response, but there's the minority response. Most recently, and so this is work done primarily by Lane Cole in Glasgow and Ronald Wright in Radwood in the Netherlands. But most recently, both in Raladino's lab at UCSF and now in Mary Angela Bozzazino's lab in Pavia, people have started to find true RNA viral sequences in the genome. By that, I don't mean there's RNA in the genome. I mean that by rare events of perhaps reverse transcription by retroviral integrase, we actually have flavivirus RNA being found in the genomes of Aedes aegypti. This is through deep sequencing projects. So what we want to do in the future, and so this idea of immune priming comes up because now you have a way of putting a sequence in. This is happening naturally in our model, and that could be affording resistance to that particular virus virtually straight away. And unlike our adaptive immune response, which of course is not inherited, this one will be inherited. So future research in our lab and ongoing research is further dissecting the whole PIWI system and the whole mosquito using antibodies and now CRISPR-Cas9 to look at each of those other genes, but focusing on 14 stars. Can we engineer immune priming against flaviviruses by inserting DNA sequences homologous to the inactive piRNA loci? And so Sally, when she was in the lab in rotation, started doing CRISPR-Cas9 against one of these piRNA loci. And now even more speculative. Can this even go to the field? Can we deliver vaccines, small DNAs to mosquitoes in the lab without the need for directly genetic engineered mosquitoes? So, is that possible? I mean, no one's thought of, we think of BT, we think of fungi, entomopathogenic fungi, and all the delivery advantages and problems that come up with that. But is there a way of developing a vaccine, which we can probably figure out in the lab in terms of delivery, but that would be applicable to the field? I'm not, that is a big idea. It's a stretch, I have to say that. But the idea of actually coming up with a molecule, a small molecule, that would allow mosquitoes to generate natural immunity as a concept of generating natural immunity in mosquitoes is not that different than what folks working on the immune system in mosquito want to do anyhow. You keep on hearing of a healthy mosquito that can kill Plasmodium or kill flavivirus. This is just a different approach that may not require genetic engineering. And I'll leave it at that. Michael Hand did most of this work from immunocytochemistry onwards and defended his PhD back in June. Jennifer worked on this in her last year as a PhD student back in 2010, 2011. Patrick Schreiner defended his genetics PhD last June, just 2 weeks before Michael. And Patrick wrote a lot of the scripts that we used to interrogate piRNA clusters that were in the background of a lot of data that you couldn't see, but you saw the outcome. Peter Arensberger was a postdoc and scientist in the lab for many years and helped us establish piRNA bioinformatics. And Rob Heiss was an SRA who at least made those plasmids. And then Lee Doss is not involved in the project, but has been in the lab during the last 3 or 4 years of Michael's work, and was great for inspiration and lots of discussions about experimental design and analysis. And Sally was a rotation student who did some injections using CRISPR-Cas9 to target piRNA loci. And we'll stop there.